All right. Wow. I've got goosebumps. That's pretty cool. But now I have to do the unenviable task of trying to introduce people that really need no introduction at all. Walter Isaacson. He is a celebrated journalist, an author, also a professor of history, University of Tulane, or Tulane University. And he's written, obviously, some of the most insightful biographies of our time, Leonardo da Vinci, Steve Jobs, Einstein, and of course, our own Jennifer Doudna. So he and Jennifer, our founder, and still one of the best, if not best, genome editing researcher in the world, are going to talk about the future of gene editing and the role IGI is going to play in that. So come on up on the stage.
Wow, this is amazing to celebrate this, and all of you who support IGI should be really, really proud. One of the things that Brad said about you is basic curiosity-driven science as a foundation for it all. But then Brad showed the picture of KJ, and then I think Fyodor even told us his first name, Kyle, and showed another picture of him. And it reminded me that what really is important is that phrase of moving from bench to bedside. In other words, the lab bench to getting it into the patient. When you founded IGI, to what extent, or you helped found IGI, to what extent was that a goal of translational science?
Well, Walter, I'll just start by saying it's a pleasure to be here with you again. And thank you to everyone here who's here with us tonight to celebrate 10 years of the IGI and the future of the IGI. When Fyodor was actually, when all of you, Ermias and Carlotta and Fyodor were speaking, I was thinking back to the moment when a student in my lab, Rachel Harwitz, and her partner, her lab mate, Blake Wiedenheft, were first researching CRISPR. And at that time, we knew it was an exciting, piece of science, kind of understanding how bacteria fight viral infection. None of us had any idea of where it would go. And that's basically the point of doing curiosity-driven research. But as you just said, Walter, one of the things that happened for us was that quite quickly after uncovering the chemistry of how molecules in bacteria are able to find and destroy viral DNA, we realized, gosh, this is going to be an incredible tool. What do we do with this? And so fortunately, I was able to speak with several of our colleagues here at Berkeley. One was Bob Tejan, who's here tonight. Another was Steve Martin, who was our dean at the time at Berkeley. And we discussed the possibility that we should form some kind of institute that would do exactly what you said, take this foundational research and turn it into real-world applications. Because even then, we could see there were opportunities. We had no idea which things we would work on, but we knew there were going to be very important things to do with the technology. And again, very fortunately, we had benefactors who decided to support this effort. And I think Julia Chow is here tonight, so Selena Chow was one of the first supporters, Mr. Li Ka-shing in Hong Kong, another supporter. And it sort of started to build from there. And again, the vision from the very beginning was we are a public university, and we want to make sure that the research we're doing gets beyond academic articles and presentations at scientific meetings to actual benefit to people.
Let me go to the basic science to start with, though. With CRISPR, so much of what's happening is advances in that technology and building upon it. And I think even the KJ thing involved things like base editing and other ways to do it. What do you see about the future of the actual science and technology of CRISPR?
Well, it's extraordinary. I think what's really exciting to me right now is that there's so many researchers that you mentioned base editing, the work of David Liu at the Broad Institute, and there's so many people like him that are developing new ideas around the fundamental technology of CRISPR that expand the toolbox, make it possible to do more and more precise manipulations of DNA that are going to make it possible in the future to do the kind of editing that you heard about tonight, sort of the visionary picture of where the science is headed. But we still need to continue to build that toolbox, and it's extraordinary to see the speed at which that is happening, not only at IGI, of course, but in many other labs around the world.
Do you still get to walk around the benches and see people who are making those breakthroughs?
Not often enough. Yeah. but I love doing it.
Yeah. They talked about AI, and of course everybody does, but the application of AI to both gene editing and to other uses of CRISPR seems much more fertile of a field than having a chat bot tell me, who's going to win the Ole Miss Tulane game or something.
I don't know. That's
Ole Miss. Tell me exactly what AI is doing these days that surprises you in helping you develop new technologies. Is it the data? Is it what type of tool is it?
I guess I would start with, I don't know if you call AlphaFold,
Yeah.
what AlphaFold is, if you call that AI, but, this is a tool
Definitely.
that was developed by Demis Hassabis and his colleagues at DeepMind Google, and was recognized with the Nobel Prize
talking
in
about it,
Chemistry
yeah.
last year, yeah. And it's something we now use every day in the lab, and I think,
So how does the knowledge of how a protein folds tie into what you can do with gene editing or
well, CRISPR is
A very
a
folded protein.
in part, at least. It's got an RNA component to it, but it's a protein.
wait, the white
And
part is the protein?
The white part is the protein.
I learned something.
No, yeah, that's very good. Now you remember from your time in the lab, right?
Jamie taught me.
And it's it's now possible to not only predict the three-dimensional structure of a protein like that, but also to predict the structures of new kinds of proteins, which is amazing, and to predict even new kinds of editors that you might want to develop for various applications. So that's going on. I think in the not too distant future, it's going to be possible to do things like say, well, I need a tool that's going to make this precise edit in this type of cell at this position with this accuracy, and be able to, predict a protein that will have that activity. I mean, it's really moving very, very fast in that direction.
And what type of data, Brad was talking about the data problems, what type of data does IGI bring to this party?
Well, I think when we think about the future of AI in biology, we keep running up against this challenge because, the exciting thing about this AlphaFold platform is that it was developed using taxpayer-funded data that came out of, probably at least 20 years of experimentation, a lot of it by graduate students and academic centers who were discovering and, kind of painstakingly, one by one, solving molecular structures of proteins such that we had a large, publicly available and very high quality database on which to train models. But unfortunately, in biology, we don't have those kinds of data sets for most things. And so at IGI and at lots of other organizations, we're grappling with that challenge. Companies are grappling with this right now too. And you may know that well, actually, I don't know if you know this, but some people here may know this, that we set up a laboratory for genomic research at UCSF with funding from a company, GSK, and that resource has been a real model for how you can start to develop the sort of CRISPR-based tool data sets that are going to allow us to predict the function of genes, not one at a time, but in terms of whole sets or pathways that lead to health or disease. I think we need more of that, and the IGI really wants to lean into kind of developing those kinds of data sets going forward.
We talked a lot when I was working on the book about the ethical concerns, and those came to the fore just as I was finishing the book. How have those changed in your mind?
Well, the ethical concerns are real, and it's something that, we still grapple with all the time, thinking about how CRISPR is going to be deployed in ways that are responsible, that are helpful to people, and not harmful. So initially, one of the very clear ethical challenges with CRISPR that came up actually quite early in the field was the possibility that it could be used in the human germline, meaning to make heritable changes in human beings. Now, the example you heard about tonight with baby KJ and the wonderful FDA-approved CRISPR cure for sickle cell disease, these are therapies that affect one individual at a time. They don't make heritable changes, but we know that the technology is capable of doing that. And in fact, there's a scientist who had announced back in 2018 that he had done exactly that. And so there's a very real opportunity or risk, really, for scientists to to really tackle this.
Let me tie these together, because one ethical concern I now have, especially listening to Fyodor, is these things are incredibly costly, and you'll have a great divide between the genetically privileged and the, it's like Huxley warned us about. But one way to change that would be to do germline editing. Does that make you think that instead of every sickle cell patient has to be a one-off, we should figure out ways to eliminate sickle cell altogether?
I think that's going to be something that we're going to grapple with this technology, because I think that day is coming, and it may be coming faster than I certainly had realized. Something that I was surprised about, actually, that's happened in the last few months, is that just here in the Bay Area, there are multiple companies that are getting started right now to focus on germline editing in humans. The idea is offering parents the opportunity to use CRISPR in embryos that they will then implant for pregnancy that will have targeted changes. It's kind of mind-blowing that that's happening, but in a way not, because we know the technology has been capable of this for a while, and it's really, I think we're now sort of at this moment where we will have to make those kinds of decisions. Personally, I think if you could eliminate a disease gene from a family that would relieve suffering, that's something that really should be considered.
We should do it probably with sickle cell Huntington's, which is almost an obvious case, it seems to me, and others. Is there some way to draw a line of where you'd say?
Well, that's the challenge, isn't it? It's how do you draw the line? Everybody might draw the line a bit differently. So I don't know how that's going to be handled, to be honest. Yeah.
You talk about what type of diseases in the future we're going to tackle. Let me just take a few topics. The first is cancer. What do you see coming up in the future there?
Really exciting. I mean, I think we're on the verge of having the ability to do targeted genome editing, precision genome alterations in human immune cells, human T cells, in the body, not ex vivo, not in the lab, but actually in patients, that will make targeted changes to their immune cells that allow those cells to fight specific types of tumors. Not just in some of the tumor types that have been targeted already with cancer immunotherapy, but going forward also in solid tumors. And I think that's going to be very, very exciting.
I always worry about tampering with the immune system. I mean, it's so fragile, and when we have COVID and other things happen, is there a way CRISPR can help us fine-tune our immune system better?
Yes, I think yes.
I feel better.
You feel better, okay. Because it's a precision technology. So, in the days before CRISPR, or even now, a lot of the types of genome manipulations that go on for cancer immunotherapy involve using viruses that integrate randomly into the genome, and that comes with risk. The great thing about CRISPR is that it can be precise, it can be targeted, it can make one change and nothing else, and that's going to be very powerful for treating disease.
What about neurodegenerative diseases? What's on the horizon there?
Well, some of my colleagues here at IGI are working on these diseases. I think that we see increasingly the knowledge for what kinds of genetic changes could affect these diseases, not only as treatments, but frankly as preventives. The challenge really has been technical, which is, how do we actually get the genome editors into the right cells, especially if we're talking about the brain. It's a very big challenge. And we've had an active program now for several years. Ross Wilson has been one of the leaders of that, to tackle this challenge head on and come up with creative new ways to get genome editors into cells of the brain where they can have a therapeutic benefit, and I think we're making real progress on that front.
I remember, I didn't see Ross, but I remember being at his lab when we were doing the book, and I got convinced that one of the bottlenecks is delivery systems, that we can just do miraculous things, but how do we get it to the right place? What's happening with delivery systems?
Lots of exciting stuff. So Ross is using a method where he's able to chemically modify not only the genome editor itself, but also smaller molecules that he co-delivers with the gene editor that help target it to particular cell types. So that's one strategy. Another approach is to make particles that are derived from viruses originally, but really don't look anything like a virus. They form a little capsule that can contain editing molecules and take them into the right kinds of cells for editing. So these are strategies that we're excited about. And then, of course, there are some great projects going on at IGI with lipid nanoparticles, which is a technology that's being widely developed, but we have some of the leaders at Berkeley that are really looking into how to do this effectively.
So how would that affect, say, vaccine delivery? Could it?
I mean, it could. You probably know that the COVID vaccine, for example, was an mRNA vaccine that was delivered with a lipid nanoparticle.
Yeah,
and
that's
in
what
fact,
made me, when you said it, think of that. And that,
yeah, and then the baby KJ therapy was also a lipid nanoparticle that went to his liver.
and so what might we do as other delivery systems besides small particles, which is, I assume, what nanoparticle means? Yeah.
Well, there's, of course, there are virus-based approaches, and again, some great research going on both at UCSF and UC Berkeley to do that, and I think at Davis as well. So we really want to try to give all of those approaches opportunities, support them, because we're sort of agnostic. We probably need all of them to do the kinds of broad-based delivery projects that are going to require innovation.
I was fascinated with what Carlotta said about the microbiome. And I always think of that as, well, do I eat enough yogurt and is my gut okay? But now I realize the microbiome is sending signals to my brain. And when you talked about neurodegenerative diseases, how could CRISPR affect the microbiome?
Well, I think there's some incredible work going on right now in our biome team, which Carlotta is part of, with the idea that we can use targeted approaches to tweak just the gene or genes in specific types of microbes. This is really a new concept. I want to make sure people really understand that here. I think Carlotta gave a wonderful introduction to this. But the idea is that instead of doing microbiological manipulations as we've done traditionally, where you essentially throw an antibiotic at somebody and it wipes out a lot of the bugs in the body at once, the idea with CRISPR is really to use precision. It's the idea that you could, in principle, make one targeted change, or maybe just a few, to one type of organism without affecting all the others.
And by a
That's
type of
very
organism,
powerful.
you mean a separate organism from us, i.e. a organism that lives in
bacterial,
us?
a microorganism that lives in us, yes. Yeah.
And so give me some examples of how that could help more than just our gut, but help our brains or Alzheimer's, something like that.
Well, I was actually at this wonderful meeting sponsored yesterday by the Kersey Foundation, and they had a number of speakers who were talking about microbiome research, and they weren't specifically talking about CRISPR, but they were pointing out that increasingly we understand in detail the connections between microbial populations in the body, which microbes are present and exactly what their genetics are, and how they affect human health. So you take that knowledge and you connect it to the ability to precisely alter the genetics of just one organism in that milieu, and you can imagine it's going to be a really powerful combination.
The huge amounts of variables in that equation you just said makes me think that's a good candidate for AI to be just taking on all the parts of our microbiome and figuring out how to.
Well, I think eventually it will be, Walter, but I think the collecting the data that will be necessary to do accurate predictions of those connections, we're a ways away from that.
Tell me about heart disease.
What do you want to know?
How are you going to save us from it?
Well, okay, so with CRISPR, I think one of the genes that gained early attention in the field of genome editing has actually been a gene encoding a protein called PCSK9. It's kind of it sounds boring, but it's actually very, very important because it's one of the proteins that helps control cholesterol levels in our bodies, and probably most people here are familiar with the fact that if you have high cholesterol, you're prone to building up plaques in arteries that can become problematic and lead to heart disease. So where does CRISPR come in? Well, the idea is if you could make protective changes to this one gene, which, by the way, one can do in the liver, which is where we do have delivery strategies today that are working, then in principle you could provide people with a one-time treatment that would go to the source, make the change, and prevent them from having to worry about heart disease in the future. It's kind of sounds like a futuristic idea, but I think it could become a reality with CRISPR.
On a broader level, in terms of policy, we've seen a whole lot of pushback against vaccines. You're very involved with the whole COVID and even ways of looking at the future of COVID. What do you see that we should be doing to try to reassure people about vaccines, and how worried are you about this?
Well, I think that question gets to a broader aspect of the IGI that I just want to mention for everyone here, and that is that, when we formed the Institute 10 plus years ago now, one of the really important ideas that I think was discussed back when I was talking about this idea with Jonathan Weissman, a colleague of ours at UCSF, and a few other people, was the fact that in addition to really advancing good science and the science that would lead to real-world applications like we're discussing, we also really needed to lean into the public policy around this because it's such a powerful technology. It's easy to misunderstand it. It's easy to be afraid of it or to, on the other hand, think it's going to cure everything tomorrow, and neither is true, right? We need to really be realistic about where it's going and how fast and what's going to happen in the future. What are the risks? What are the benefits? And so I've been really proud of our public impact team at the IGI, now led by Manar, who I think is here tonight. And we're really delighted that you are leading our team in this regard because we have to be an institute that provides real data. We want to provide facts. We want to be a trusted source of information. As you know, increasingly people turn to TikTok videos or, Instagram posts and things like that for their information about their health. And, a lot of that is not really fact-based. So we want to make sure that we're an organization that people can come to when they want to find out what are the data, what are the facts, and then they can make an informed decision.
About a year ago, you were in New Orleans for something we did, and Francis Collins was there, and we all had a dinner, and there was a discussion about this very topic, which is the public's misinformation about science, now fear of science, being stoked up. I know what you're doing at IGI, and it's good, but is there some national movement we can have, something that people like you, Francis Collins, and others can try to do, to once again have people appreciate science, including the things we saw today? I mean, I think if you could just have pictures of KJ everywhere, you could do this. Yeah.
That would help, yeah.
Yeah.
Look, I think this is just my own personal experience. I've found that, when people have a personal situation where they can benefit from science or where they have benefited from it, where they can relate to it, then they lean into it. And I'll just give you one really quick example that really brought this home to me in an interesting way. So this is probably more than 10 years ago when I had been invited to Washington with George Church from Harvard to talk about genome editing. It was very early in the field, but already it was clear that this was going to be a really impactful technology that our legislators need to be aware of. And I was, so we were doing a small kind of round table with mostly staffers, and then we were told, well, we're going to have a dinner with some of the representatives in the House, and they showed me the seating chart, and I realized I was sitting right across the table from a new member of the House of Representatives who was a staunch, very conservative Republican, and I thought, oh dear, this is what am I going to say to this person about CRISPR? And what happened was very interesting, and that is we sat down at the table, and she leaned across the table, and she said, look, I want to tell you a story. She said, I was pregnant last year, and during my pregnancy, it was discovered that my child had a rare defect in the kidney that was really difficult to treat, and it made for a very difficult pregnancy. It was terrifying. The child was born. They had to have, complex therapies. She said, I just want you to know that if your technology had been available for my child, I would use it. It was very, very interesting.
You said, make it connect, make it better. There's probably some questions out there, and we have a few minutes or so, and I was told that everybody would be eager to do questions. So, number one, yeah. Oh, Brad, okay.
I'm just the messenger. Do we have questions from the audience? We got 5 or 10 minutes.
All right, well, everybody, oh, here we have a hand, finally.
have a question.
Undergraduates are much more, they all have their hands up.
It's a great pleasure to be here. My name is Sergio Ferreira. I'm the scientific director for Pioneer Science Initiative in Brazil. And we've been partnering with the IGI for a few years now, and it's a real privilege to be here today. I think we are all super excited and super looking forward to the expansion of these techniques, as you and Fyodor's vision have been telling us that this will be the goal, to open this up to the whole world and make it accessible and affordable. On the other hand, I think that it's clear also that, at least for a while, companies and biotechs will play a role in this. And I've been reading about reduced funds, not only from governmental sources, but also from venture capital and things, and companies laying off personnel. So how do you think this is going to affect the field? Is this going to be important? How are we moving forward?
Boy, I wish there was a short answer to that question. How long do we have?
As long as you want.
Oh boy, look, it's a worrying time for all of those reasons. First of all, as you know from the introductory comments from Brad and others, that the IGI partners with companies all the time. We're very proud of all the spin-outs that we've had. We're proud of our entrepreneurial programs where we support people that want to found companies. This is a key element to scaling the kinds of things we're doing at IGI. We're never going to be able to scale the things you heard about, right? We're going to need companies and investors to make that possible. And I do worry about that ecosystem right now. I think it's been highly successful up until now in the US, and especially maybe in certain areas of the US, especially the east and west coasts. But it's at risk, because if we cut down funding for fundamental research of the type that you're hearing about tonight, then you cut off that pipeline, and it makes it very difficult for those starting companies or even established pharmaceutical companies and everybody in between to get access to the kind of innovation that really only comes from the kind of high risk, but ultimately in some cases high benefit science that the government has traditionally supported. So I think increasingly we're going to see that ecosystem at risk. I think, to the extent that any of you have influence in Washington, it would be good to communicate this. I've had some very interesting conversations one-on-one with representatives from California around the state, not just those that represent my district, from both sides of the aisle, Democrats and Republicans, and one-on-one, to a T, all of them have said the right things in terms of, we want to support science, we know it's important, et cetera. And so far, you may have noticed that in the budget negotiations, those, the Congress is supporting NIH funding levels at, sort of the current levels. But will that really hold going forward? I think only if we all speak out.
We actually have Google DeepMind representatives here.
Yeah, no, I was about to say we had AlphaFold here.
We're so thrilled to be partnering with you all.
Well, why don't you introduce yourself? 'Cause I know
My name
who
is
you are.
Dorothy Chow, and I lead public engagement at Google DeepMind, and really pleased to be partnering with the IGI in a number of initiatives. Jennifer, my question for you is, if you look at the next 5 years of what's happening in AI, what biological questions do you think are the most tractable? And then which do you think will resist computational shortcuts, especially when I think what we see predicted in silico may not be validated in vivo? And I'd love to hear more about that from your perspective.
Yeah, no, Dorothy, it's a great question, and something I think about a lot. I don't know if I have a great answer yet, because I'm still thinking about it, but I'll tell you what I'm thinking right now. So I think as we discussed briefly tonight, the opportunity to predict molecular structures, to do things that some of your colleagues at DeepMind, and of course many others, are doing quite effectively right now, which is to predict small molecules that can bind to proteins and have an impact on proteins. I think that is advancing very quickly, and it's really exciting, and it's technology that's being used by both academic and corporate groups at the moment, and will continue to. I think what's hard to do, and you alluded to this, is that it's really hard once you start to get to the level of an organism to predict how molecular interactions are going to affect a pathway, and much less, the behavior of a person, let's say, is just really hard. And this is something that all of us that are biologists are familiar with. It's just a big challenge. It's the reason that a lot of clinical trials don't work, for example, right? You have a lot of good data and companies go forward in good faith thinking, we've got great data, but then you get into a person and, things go haywire. So I think that's going to continue to be a real challenge. I'm actually quite excited about approaches that integrate the sort of molecular work that we're talking about here with more organismal level effects. For example, being able to, and a number of companies are doing this, including one that I'm on the board of, Tempus AI, but there's many others too, that are looking into how you take patient data and health records that are anonymized, where you have outcomes for treatments with certain types of therapies, and you can start to very accurately predict, based on somebody's, let's say a tumor genetic profile, for example, which therapeutics they're going to respond to effectively. I think that is a really exciting future direction for AI that I'm very excited about. And back to a more kind of fundamental molecular level, I think being able to do that type of prediction for cell and tissue behaviors is also coming along as well, which I'm very excited about.
In fact, I think Demis Hassabis is working on what you would call a virtual cell or a simulated cell, so that you could do the experiments in the virtual cell, which would help really accelerate
Science, yeah.
All right, I've moved to the back now. I
We
was
can't
I was
see you.
remiss. I'm back here. I was remiss because I did not recognize our board members that are here today, and our chair of the board, Steve Altschuler, is right here. Thank you, Steve.
How are you?
I've got Kathy High right here in front of him, and then Bob Tijan. I think Bob is Tijan's here as well. Thank you, guys. Now, I was supposed to give Steve a chance to ask a question. If he wanted to ask a question, Steve, are you up for that? Do you want to? All right, I'm going to pass it through to you.
And Steve, thanks for what you're doing.
You're welcome. So, we talked about therapy for devastating disease in children. And I think when you introduce these type of therapies, you want to make sure the first patients, the first indications you go after, you have success. Because if you have a problem, it can derail all the therapies. And, having lived this experience with Kathy many years ago in, kind of the earliest parts of gene therapy, it's really critical to think about what indications and how you choose patients. And I was wondering what your thoughts are on that.
Yeah, it's a really important point, Steve, and a question. Something we think about a lot. We're sort of keenly aware that we have to be very thoughtful about the patients that are selected for, especially for this new center for rare disease that we're developing at UCSF. Fortunately, we've got some wonderful clinicians. Some of them are here. Matt Kahn was mentioned earlier, and of course Sam Hoggood is a pediatrician, and all of their colleagues at UCSF who we're working with to make sure that we really are thoughtful about how we vet patients for the trials that we want to do. And I think we're also benefiting from the fact that now, there is a CRISPR therapy that's FDA approved for sickle cell disease. So there's some, increasing knowledge as people are treated with that therapy for how they respond. And as you probably also know, there are many, many clinical trials with CRISPR that are ongoing at companies. So we're constantly seeing papers published, data posted from these trials that continues to advance our knowledge base for how these therapies are being received and how effective they are. So I think we're just going to, in real time, we're going to continue to evaluate these patients and use the best knowledge that we have to make those decisions.
So one last question, and it's actually a question that a far better interviewer than I am asked you a couple days ago, my friend Jeff Goldberg. And he asked you a question about if you had just a few private moments with President Trump, other than showing the picture of KJ, which would solve everything, what would you tell him? And I think that's relevant too, because for the donors in this room, it is, what do you want to tell him and everybody about investment in this?
Well, I guess just two things. I would say that for every dollar of NIH money that is invested, we get at least $2.50 back. That's a good deal.
The art of the deal.
And secondly, I want to keep America number one.
All right, IGI is in the fore of doing that. Thank you very much.
All right. I can't think of a better way to kick off the next decade. That was fantastic. All right, we have the room here until after 7:30, so please mingle, talk with the scientists that are here. The whole idea is just to be here and celebrate the IGI and the work that we do. And again, there's signed books, so if you have not picked up, I don't know how many doubly signed Codebreaker books there are between Jennifer and Walter.
It's the only time I've ever seen, except for maybe a few in New Orleans, in which the book has both our...
Yeah. So I don't want to see him online. So don't sell these collector items. So again, I just want to thank everybody just for what you have done to help make the IGI what it is, and I invite everybody to be part of our next decade. Okay? Thank you.